Hi there. Okay, I think we've got most people now joining the meeting. So firstly, let me welcome you to the session and thank you for joining the webinar this afternoon. Before we start, I just want to touch on some housekeeping. If you have any technical issues, then please send a short message in the question box at the right-hand side of your screen. During the webinar, RSSL would like to invite you to ask questions also using the question box on the right-hand side of the screen. At the end of the webinar, time permitting, we'll try and answer some of those questions if possible. If we don't get the opportunity to do so during the session, then we'll endeavour to answer them offline and come back to you with responses. So today's webinar is on disinfectant validation, and this is part one of a two-part series with Dr. Tim Sandler and myself, Nikki Boyle. So a little bit about me. My name is Nikki Boyle. I'm the Microbial and Sterility Account Manager at RSSL. My contact details are there, so should you require any of these services, please feel free to reach out to me. A little bit about RSSL. We are a CRO specialising in analytical chemistry. We are about 40 minutes from London, based on the Reading University campus. We are both MHRA and FDA approved and ISO accredited, and we've been in the market for in excess of 35 years. We pride ourselves on being flexible and agile across quality within the whole organisation. And these are some of the services that we support our pharmaceutical biotech clients with. So environmental monitoring, microbial identification, particulate control, sterility assurance, extractables and leachables, raw material testing, including bioburden and endotoxin, cleaning and disinfectant validation, which is obviously today's topic, and also vial and stopper testing and container integrity, so very much around the medical device piece. And we also offer training and consultancy services if required. So I would like to introduce today's speaker. This is Dr. Tim Sandle. Tim has over 30 years of experience in the field of pharmaceutical microbiology. He is a member of several editorial boards, also extensively published, multi-award winning, is actively working in the pharmaceutical industry, currently heading up GXP compliance and sterility assurance at Kendrion Biopharma, and he's also an associate professor at UCL. So I will hand over to Tim.
Hey, thank you very much, Nikki. I shall just switch my slides over. A second. Okay, just check. Can you see my slides okay? Okay, right. Well, welcome everybody. I was going to turn my camera off in order to preserve the bandwidth, and I'll turn it back on when we move to the Q&A. So, as Nikki said, this is a two-part series. We're looking at two different dimensions of disinfectant efficacy testing. In this one, we're going to look at the selection of disinfectants and the basic test methods. And then in the second part, we're going to move on to the field trial, how we're actually proving that our disinfectant works in real life. So in this webinar, we're going to be looking at how we define and approve disinfectant, how we should select a disinfectant, what are the regulations that we need to take note of, what do we do in terms of test standards, and should we slavishly follow those test standards, or do we need to adapt those test standards according to the requirements of a pharmaceutical facility? We'll also be looking at briefly looking at the test, which is not of huge value, but we need to note it, and then more detail on surface testing. I will also touch briefly on hand disinfectants, hand rubs, hand sanitizers, and wrapping up by saying what are some of the key things that we would need to note if we get a regulatory inspection that's looking at how we have introduced and qualified our disinfectants. So let's begin by looking at the approval of disinfectants. So in most countries around the world, there is a legal requirement to prove that disinfectants that are put on the market are equally safe, or at least the safety measures are well known and appropriate preventative measures can be put in place to protect public health, and also that they are effective. And that's achieved through a basic level of testing and then registration with the appropriate authority. So, for example, to sell a disinfectant within the European Union, then it needs to meet the biocidal products regulation. And similarly, in the US, it would need to meet the federal insecticide, fungicide, and rodenticide act as well. So, and the important thing is, although a disinfectant might be registered and it will have had an element of testing, this isn't sufficient to provide the end user, i.e. yourselves, with the degree of assurance regarding the performance of the product. So it gives a basic level, this disinfectant has a certain level of activity. It doesn't necessarily mean that a given disinfectant is suitable for use within a pharmaceutical clean room or a healthcare facility. So we need to go through a process of selecting disinfectants. So before undertaking qualification, we need to make sure that we're using the right disinfectants as required by the end user. So there's no point going through a lengthy and often proves relatively expensive qualification process if we're going to end up with products that are not user suitable. So we need to consider whether we're using disinfectants that have different modes of action. So by that means that are the disinfectants able to kill the microbial cells in different ways? And this is on the basis that certainly within MHRA and European Union GMPs, that we need to be rotating between two different disinfectants, and there's no value in rotating between two disinfectants that have the same mode of action. We also need to consider whether the disinfectants have sufficient spectrum of activity. So by that we mean the range of different types of microorganisms that disinfectant is at least theoretically effective against. We also need to consider whether we are using a sporicide. So we can either use two different disinfectants and then have a sporicide that we use at periodic intervals, or we can rotate between a standard disinfectant and a sporicidal disinfectant. And again, that's central to Annex 1 of EU GMP. We also need to consider whether our disinfectants are compatible with the detergents that we're using. So we'll touch briefly on residues, but we need to assume that there might be some residue activity. Therefore, we need to be sure that the disinfectant and detergents are compatible. And that similarly goes for the two different disinfectants that we will be selecting as well. We also need to think about residues, as I've mentioned. We also need to make sure that the contact times are realistic. So is there any value in selecting a disinfectant with a contact time of 60 minutes if that's not going to fit into a busy manufacturing schedule? We also need to weigh up the likelihood of material damage, and as a general rule, disinfectants that are chlorine-based have the potential to cause material damage, so we need to consider how we're going to remove residual disinfectant. We also need to think about what formats the disinfectants are available in. Again, there's no point qualifying a disinfectant that's only available in a concentrate form if we also wanted it available as a trigger spray and as a pre-saturated wipe and so on. And we also need to ensure that the vendor we're purchasing from is suitable. So can our disinfectant be at least lot controlled? What are the safety requirements? So again, if we're going to introduce a disinfectant with a particular pungent smell, how are operators going to react to that? And do we have any specific PPE requirements? So will we require operators to wear respirators, for example? And also we need to consider how we can safely dispose of the disinfectant after use and meet environmental legislation, because we don't want to be fined by the Environment Agency for pouring tons and tons of unauthorized chemicals into the public drainage system, as an example. And we also need to be mindful of regulatory expectations. So we mustn't lose sight that the testing that we're going to undertake is performed to demonstrate that our disinfectants are fit for purpose. And that is that they provide a sufficient level of reduction in the viability of the types of microorganisms that we expect to encounter on inanimate surfaces within clean rooms and healthcare facilities. And we also need to ensure that our testing is suitably robust, in that we are able to stop the disinfectant activity and adequately neutralize it, so we're seeing what's happening in the practical consequence. So we see references here to the PICS guide and also to the FDA guidance on aseptic processing, and also to EU GMP Annex 15, which is the annex for validation, but also EU GMP Annex 1, which is of great consequence for sterile product manufacturing facilities. And that's very clear that disinfectants and the disinfection process, and the emphasis upon the process will also link to the second part of this webinar, is validated. And we need to account for the manner in which we're going to use these disinfectants. So again, there's no point having a theoretical assumption in a test tube if that quantity is not then available on a mop head. And also the types of surface materials, because we can get a wide range of different outcomes for different surfaces. So that's the regulatory background, but what the regulations are not doing is saying you have to use standard X, Y, Z. There are different global approaches to disinfectant validation, and there is no global harmonization. And in theory, there's no right or wrong approach. But like any validation exercise, we need to have clear criteria, and we need to work out how we're going to demonstrate the criteria and how we justify the choices that we have made. So some of the more common guidances, there's the United States Pharmacopoeia chapter titled Antiseptics and Disinfectants, chapter 1072, which is a guidance chapter. There are, in the US, there are two sets of standards. There's the ones from the chemistry series of standards, the AOAC. These, in my opinion, are not brilliant, and I wouldn't recommend following them. The ASTM standards are of far more value, but there are also the European standards, the European norms that come under the CEN. And there are differences between all of these different standards in terms of the methodology, etc., etc. And it might be that none of these are absolutely suitable. We might need to make certain adaptations in terms of, what is the microbial population that we're likely to see in a clean room in terms of numbers and types of microorganisms. And there's other things as well that we need to account for, like drying times, which we'll also come on to. So looking at the European standards, These are divided into different steps or phases, and if you kind of do a search for European standards, there's a number out there, a number that are not applicable to pharmaceuticals and healthcare. There's some for veterinary settings, for example. So boiling down those that are most suitable for use and adaptation. We have some for basic activity, and all of those ones on the slide are the kind of basic ones, whether they're qualitative or quantitative. And these are the types of tests that we would expect any reputable manufacturer of a disinfectant to have carried out. So these are not ones that are normally replicated, but we'll come on to that in a few minutes. And these standards continue into surface testing. The surface testing is what is of fundamental importance, and the service testing is often what only a facility itself can do or contract out to to a contract testing company. So these are now the two kind of key tests that we'll come on to, as a general one, and then one that's looking at wiping application. Another thing that these tests share is that they have a standard panel of microorganisms. And these microorganisms are there so that, in theory, you could go to any laboratory that claims to have performed testing against the standard and make a comparison. Whether the organisms are partly or wholly suitable for your particular application is again a choice that needs to be made, and certainly some of those organisms are probably suitable, like Staphylococcus and a representative filamentous fungus, but we need to make choices in between that, and we'll come on to that as we work through. And we also need to consider how we're going to approach sporicidal disinfectants, because the inference there is that all of those organisms listed are in the vegetative state. So, like any other validation exercise, we need to start with some form of validation master plan. Incumbent upon us to have a reasoned and justified approach to our disinfection validation studies. And it might be that applying a quality risk management approach is suitable, because we need to start making choices about what we want to do in terms of surface types and neutralizers, and also, holding times for disinfectants, and what we want to achieve in the field trial, which will be the subject of the next webinar. So we need to have all these kind of sewn together in one document, and that requires us to make a series of choices. So to my mind, each one of the things on the circle on the screen need to be included in the master protocol or supporting rationale. So we need to decide which microorganisms do we think are representative of our clean rooms that we wish to evaluate against our selected disinfectants. We also need to decide what types of reductions in microbial counts we would expect to see based on what we might find within the environment. And we also need to decide the level of reduction as expressed in logarithms that would be suitable. So are we going for a 6 log reduction, as some of the standards might infer, or are we going to accept a 2 or 3 log reduction, which might be more typical of the bioburden within the clean room? If we're going to challenge our disinfectants with a soiling substance, then what are we going to select for that? Are we going to adopt what the standard recommends, which is bovine serum albumin, or something that might be representative of our own product residues. We also need to select a suitable neutralizer, not only for our efficacy testing, but then what ends up in our contact plates. We need to decide on how many surfaces we're going to evaluate and what is a representative surface. The formats which I've discussed, and whether we would need to separately evaluate the type of solution of disinfectant that we might be applying, sort of mop and bucket approach to what might be embedded within a wipe. Concentration of disinfectant. I mean, largely we'll probably start with the manufacturer's recommended concentration, but that may or may not be wholly suitable. And then the contact time, how long are we going to wait for the disinfectant activity to last for? So you can see it becomes you know, a relatively complex series of decisions that we need to make. And if we don't do all that thinking up front, then we could either end up with the wrong things, or we could get muddled up partway through our qualification exercise. Right, so now we're going to have a look at the different test steps. So I mentioned the suspension test, and this is essentially a test tube activity. And it's divided into more of a qualitative test and then a quantitative test with soiling. And essentially all these is that you have your disinfectant in a test tube at the concentration that you think is going to be effective. You challenge a microorganism into that. You'd hold it for the contact time, and then you would stop the disinfectant activity by adding some form of neutralization step. So these are of limited value, but they can help if you have lots of different disinfectants you want to select from and you want to whittle those down, or you want to get a rough idea of what the appropriate concentration is, or the minimum inhibitory concentration. But ordinarily these are kind of tests that a manufacturer should be performing. And the reason they're of limited value is because it's not common for microorganisms to be in the planktonic state or free floating. So most microorganisms, certainly within a clean room environment, are attached to a surface. And the longer a microorganism is attached to a surface, the harder it becomes to remove. And we may get added complications with biofilm formation. So it's kind of a bit of an artificial construct, but it's a useful screening test. So, as I mentioned, we would challenge individual organisms with individual solutions of the disinfectant at our chosen concentration for a particular time. We'd stop the reaction with a neutralizer. And also what we need to note is that when testing, because we have to do a dilution step, the actual concentration of a ready-to-use disinfectant is actually tested at 80% of its full strength. So either we accept that, or we would need to perform a adjustment to the concentration. But of far greater importance is surface testing. So in order to demonstrate compliance with predetermined acceptance criteria as expressed in log reduction, we need to use some form of quantitative method and take into account the types of surfaces that we would find in our facility. And we need to make a choice about whether or not we include mechanical action, or we might wish to evaluate how well a disinfectant applied to a surface works in itself, and then how well it works if we apply mechanical action. So mechanical action is designed to replicate the activity of mopping or wiping, because the application of pressure can help us get the disinfectant to make more chance of making contact with the microbial cell. And that's particularly important with surfaces that are becoming older, might be slightly scratched or whatever. But also the act of wiping, the application of pressure, can also help to disassociate microorganisms from the surface. And going back to the thing I was saying about microorganisms that are not attached to surfaces, they're far easier to kill than microorganisms that are bound to surfaces. And as we embark on surface testing, there are some things we need to consider. And with the standards, they do have very, very high starting inocular. So they're talking about challenging with 1 million or 10 million microbial cells. And they don't account for mechanical action, and sometimes they're just based on, a most probable number of survivors. So we might wish to go with lower log reductions, and we might want to go with lowering starting inocular, which we'll have a look at in a moment. Another thing about surface testing is that it does require a lot of skill and precision, and often these are laboratory techniques that are not part of the normal day-to-day range of skills found in QC microbiology, which is again a reason why a lot of laboratories would opt to contract out. Another thing with the standards is that essentially they use stainless steel, or in the case of the US ones, porcelain, either discs, coupons, cylinders, however they're described. And that's because stainless steel is the kind of the base material for evaluation. Now, while stainless steel is often found in abundance in GMP areas, we're going to have a lot of other different types of surfaces. We're going to have vinyl, we're going to have glass, we're going to have acrylic and other types of materials. And it's a common regulatory finding, one, if a facility hasn't carried out surface disinfection, but also two, if the surfaces are not representative, or there's no evidence of a rationale that has said, I've picked surfaces of particular types. And this is important because different microorganisms have different levels of attachment, so there's different physicochemical forces at play, and different levels of resistance, and there's also different levels of absorbency and different levels of cleanability, and so on. And plus there's also things like surface finishes that can affect the ease or otherwise of disinfection. So there's factors to weigh up, but equally it's also important to take a nuanced approach. So if a given disinfectant is not used on a particular surface, then there's no point including it in its validation. So if we have our two main rotational disinfectants and we're also using IPA, but we're only using IPA on gloved hands, there's no point including that IPA within the surface studies. We're just adding to the cost. Another thing is where we have surface materials that are very similar, but we might have things of different grades. So if you take stainless steel, it'd be common that product contact surfaces have 316L stainless steel, but other forms of stainless steel are 304. It's probably not worth doing both types of stainless steel. But one of the decisions to be made is, do you do the 316 stainless steel because it's product contact, or is the 304 a tougher challenge because it happens to be a slightly rougher form of stainless steel? So we have to make these choices, and we need to come up with a list. And typically it's 4 to 6 surfaces that are selected. And in my experience, if you kind of show a regulator that, you know, you might have 10 different surfaces in your facility, but there's a few ones that aren't, you know, they're not near anything significant, they're not particularly commonplace, they're not going to affect environmental or product quality, you can sort of skip those and you kind of boil down to your key surfaces. And as long as you then demonstrate that the in-use disinfectants are effective, or the new disinfectant you're wanting to introduce, then that's normally sufficient. And generally you'll be including stainless steel, acrylic, glass, vinyl, and different types of plastic. The other thing I've mentioned, which is an important choice, is the neutralizer. So it's important that the test method that we're deploying does not have a deleterious effect on microbial recovery. So we need to show that microorganisms are not inactivated as a result of the test method, rather than exposure to the disinfectant. So that means that the neutralizer that we deploy to stop the activity mustn't be toxic itself to the microorganisms. So we will need to prove a separate study to show that the neutralizer is not toxic. But we also need to show that the neutralizer is effective. So that means it's capable of quenching the activity of the disinfectant, so that we can achieve neutralization at our targeted contact time. So without knowing these two factors, we can't really determine whether the disinfectant is effective or not. So it's an important series of related tests. Another choice that we have to weigh up is with the test microorganisms. So simply using the organisms that are listed, say, in the European standards would be open to criticism if these organisms are not representative of the microbiota that we would find within the clean room facility. So often, if we use the standard list, we would need to supplement that by widening the test panel. And if we widen the test panel, we can either use environmental isolates, or we can use type cultures. And again, there's differences in approaches, and often there's different regulatory expectations. So regulators like to see the inclusion of environmental isolates. This is partly under, in my view, a misbelief that these organisms are somehow more resistant than laboratory cultures, and maybe they are in the clean room, but there's no proven way to replicate that resistance once they're in the laboratory. But however that's done, the panel often needs to be expanded so it is representative of what you're commonly finding within the facility. So if you take the fact that Micrococcus luteus is generally, according to most surveys, the universally most common organism, that probably should be included. Now, some may make the argument that Staphylococcus is very similar, and to a degree it is, but Micrococcus luteus's ability to survive for prolonged periods in the clean room is almost unique. It certainly is one of the hardiest non-spore forming organisms. So what's the panel going to be made of is a key choice. We also need to consider how we are going to cultivate and look after those organisms to avoid phenotypic changes taking place. So we need to understand that as well. And if our disinfectant has sporicidal properties, or the manufacturer says it has sporicidal properties, we need to include organisms in the spore state. So we need to be careful here that we don't just use Bacillus subtilis without knowing that that Bacillus subtilis is in a spore form. Otherwise, we just use vegetative cells. So we need to understand, are we going to buy in spores, or if we're going to create our own spores, how do we know that we've got sufficient numbers of spores and it's sufficiently free from vegetative spores? And there's a further complication in that the European standards point out that fungal spores are found in different forms and stages. So the recommendation there is that fungal spores are in the state where they're called spiny spores, which relates to their morphological characteristics, but it's been proven that these spores are more resistant. So where we're using Aspergillus brasiliensis, then we would need to get hold of a spore crop that has them in the spiny spore state. Contact time is another important consideration. So the contact time, as I'm sure you're aware, is the time that an organism is exposed to the antimicrobial action of the disinfectant. So it means that the disinfectant has to reach the microbial cell wall and exert its antimicrobial effect. So, as I said earlier, how long a contact time do we want? It's often the case that the contact times for sporicidal disinfectants are longer, but even for non-sporicidal disinfectants, to achieve fungicidal kill is often slightly longer than bactericidal, and where we're using a sporicide, that is typically longer than killing of vegetative cells. So we need to account for that and how long is suitable for the manufacturing. We also need to consider that some disinfectants will evaporate faster than others, so those that have greater volatility. That a greater evaporation will occur on warm surfaces, or where we've got low humidity, or also where we've got high airflow. So using a disinfectant in a grade D clean room is very different to using a disinfectant under grade A conditions, where we've got very, very fast and very, very dry air coming down. And the reason why that's important is that to be effective, the surface needs to remain wet. Now, I know there's some literature that says if a disinfectant dries, there's a degree of residual activity, but that's relatively limited. So if we apply a disinfectant, it's supposed to have a 5-minute contact time. If it dries in 3 minutes, we need to do a reapplication. So we need to weigh up again whether that's suitable and practical for our industrial setting. Also mentioned the mechanical action. So there is a European standard for wipes as well. So that decision needs to be made whether we're going to look at the disinfectant on a surface without wiping and with wiping, or just with wiping. If we're going to go just with wiping, we need to be sure that whatever we've done in the validation exercise is reflective of what we're going to do when we're using that in real life. With acceptance criteria, what's in a number of the standards is too high for what we'd see in a clean room. So particularly when we come to sporicidal disinfectants, many people struggle. If you start saying off, I must achieve a 6 log reduction, and you're actually struggling to get that for a sporicide, you know, if you've got a facility where you've got 1 million spores already, then you're in a really bad situation, because the permitted bioburden on a surface in grade D, according to EU GMP Annex 1, is 50 CFU. So we shouldn't really be having challenge is, more than 50, more than 100. So as a general rule, as long as you've got disinfectants that can achieve sort of 3 log reduction for vegetative organisms and a 2 log reduction for organisms in the spore state, then you've probably got a disinfectant that's going to be suitable for most pharmaceutical and healthcare environments. However, there is one caveat, and that is, even if you're targeting a smaller log reduction, it's more precise to go from a higher level to a medium level. So if you challenged with 10 million cells and you wanted a 3 log reduction, so you're targeting 100,000 survivors, that's easier to measure and to quantify than if you were trying to start off with 100,000 cells and you wanted to go down to 1 cell. And that's because it becomes more and more difficult to measure the smaller the number, and there's a whole range of issues around the accuracy of plate counting and things that won't go into, but it can be dangerous, particularly if you set different criteria between replicates and things. The smaller the number you're going for, the more difficult it can get to achieve that. So there are variables that we need to consider and bear in mind when we are taking these disinfectants really into the clean room. So the first one is that disinfectants are less effective against high numbers of organisms compared to low numbers of organisms. So we need to bear that in mind. Stressed organisms might be more susceptible or less susceptible than the organisms that we're using in the laboratory. And also we will be hopefully applying our disinfectants with wiping activities. Hopefully we've moved beyond simply just spraying and hoping for the best. So we need to factor in all those, and these are things that can sort of help to shape our understanding of setting the criteria and the data that we end up with. We also need to consider what water we're going to use where we're diluting our disinfectants. So the standards talk about using water of standard hardness, and that is very clear. That is a worst case because the presence of minerals can, to a degree, deactivate our disinfectants. But if we're in a pharmaceutical setting, then all the water that we will be using will be purified, maybe water for injection. It's gone through some softening process. So again, that's something we should factor in to make sure our test method is reflective of what we're going to actually be doing in reality. We also need to consider temperature. So the standards tend to recommend a temperature of 20 degrees Celsius plus or minus 1 degree, although more recent revisions that they're kind of a little bit broader with their temperature recommendations. But there is something called the Q10, which is a quotient. It's a universal rule for disinfectants, and it essentially says that for every 10 degrees increase in temperature, the rate of reaction is halved, so our contact time. But conversely, for every 10 degrees decrease in temperature, the rate of reaction doubles. So if we've done all of our laboratory testing at 20 degrees, and then we want to use that disinfectant in a cold room, then our contact time probably needs to double. And that's either something that we just say, okay, the laws of chemistry say we need to double it, or it might be something that we want to try and evaluate that under colder conditions. And then I mentioned earlier about soiling as well. So unless we've got exceptionally good cleaning and good application of detergents, our clean room surfaces will probably have residues of something behind, protein, grease, something like that. So it's important that in our disinfectant testing that we at least understand what the effect of trace residues of soil are. The standards recommend bovine serum albumin, and that might be a worst case, or there might be something more applicable. So you need to weigh up when we test our disinfectants, what type of soiling and what degree of soiling do we wish to have in there to simulate that process. We can also face problems when we're using certain organisms that maintaining the viability of those organisms can be problematic. So that's particularly the case of if we're using a Pseudomonas, particularly Pseudomonas aeruginosa, that it's very sensitive to drying. So if we face that, we might need either to start with higher concentrations, or where that becomes particularly problematic with bovine serum albumin, then some practitioners recommend using a different soiling substance like milk powder. We also need to factor in that disinfectants might leave residues, so that when the people who come around and perform environmental monitoring are using contact plates, we need to make sure that those contact plates contain a neutralizer. But it's important that it's the right neutralizer, that it's there's no point having done surface testing with neutralizer A and then our contact plates contain neutralizer B. We need to know that the neutralizer is the same and it has that same level of neutralizing ability. Otherwise, we are undermining our environmental monitoring data. Okay, so just briefly on hand hygiene, that there are two European that require the addition of water, and hand rubs which don't have the addition of water. So both of these hand products can be directed against transient skin microorganisms. Again, this is generally an evaluation that the provider of the disinfectant should be doing, because these are notoriously difficult for any facility to attempt to use. You need 18 to 22 volunteers, and you have to use a attenuated strain of E. coli in a soup. So it's more common to accept the manufacturer's data. Another factor is with disinfectant compliance. So if we vary from a standard, then it's important that we don't claim that we're absolutely following that standard. So we can, it's fine to modify a method, but we just need to be very clear what we're calling that particular standard that we're using and what we're actually attributing our testing to. The other consideration that we need to make is with how we're approaching testing as well. So as I mentioned before, this isn't a routine activity, and it's often challenging for many laboratories. And I'm not here necessarily to say it's too difficult to do, but it is something that you sometimes need to consider the services of a contract test laboratory. And I know that RSSL do offer this service. Okay, so this brings the presentation to a close. So it's really important to have undertaken disinfectant qualification. It's important to understand how our disinfectants compare to our contamination control strategy, how we've qualified them, how this works in practice. The surface testing is the most meaningful to conduct, but whatever we do has to be supported by a field trial. The field trial is the ultimate proof of the suitability of that disinfectant. So they're the key points I want to get across. We'll deal with the field trial in the next presentation, but I can hand over for Q&A.
Thank you, Tim, for that information and comprehensive review of disinfectants. We have some questions that have come in, so I'd like to take a few minutes of the remaining time to sort of go over those. So just let me share my screen very quickly. So, great. Technology. Yeah, so a couple of questions that I'd like to throw your way, if that's okay.
Sure.
How often would you suggest that organisations validate their methods?
I think you can kind of approach this as like a one-off activity.
Okay.
But then again, if you see a shift, and one of the things I'm a great advocate of is trending the microorganisms that you find within your clean room. So if you start to see shifts in the types of microorganisms, then there may be a case to challenge those. Or as if it's quite common, like manufacturing staff want to change the frequencies, or they want to change the type of mops they use, and all those sorts of things. So it's whenever variables alter, and then using a change management system is useful.
That's really helpful. Another one, I think there's sort of MHRA are quite hot at the moment. Will regulators accept suspension tests?
No, from my experience, no. You really need surface data, and these days, often when you have an inspection, they say, "Bring me your surface data." So if you kind of march in with suspension testing, that you probably get a finding.
Okay, no, that's very helpful. Another one, how would you select isolates for disinfectant challenge?
So I start off looking at the panel, then I would look at the outcomes of environmental monitoring data. So one, it's important that you've done a reasonable number of identifications. So not just simply always just IDing everything over action level. You've got a broad representation of, particularly if you've got a facility with grade D, C, B and A areas, that you're looking across that, and you're accounting for the particular problematic and more resistant organisms. So you need to be able to defend, to say, I've used 6 organisms in my disinfectant efficacy testing, and here you go, here's my profiling, and I believe that the 6 I've used are broadly representative and suitably done a suitable challenge of what's really out there. You've always got to keep connecting this validation to the real world, otherwise it all becomes pointless.
Absolutely. Okay, well, that's really helpful. One final one, unless the audience have got anything they want to pop into chat, and I'm going to try and get the pronunciation of this word correct. Which spiricide is the safest to use?
There isn't really one, actually. So it's answers the question that comes up. So sporicidal disinfectants are designed to kill all life forms.
Okay.
Therefore, they're all nasty chemicals. So often people will say that's really pungent, or I have to wear a respirator, or anything else. But some of the things like hypochlorous acid are probably a little bit safer. But these are all quite nasty and aggressive chemicals because they're designed to wipe out everything.
Thank you for that. So just let me stop sharing for a moment. And just to check if we have, I can't see any other questions coming in. So I think that takes us to the end of our webinar for today. So I'd just like to sort of round off by saying thank you for everyone who's attended the webinar, and Tim for your kind of overview and insight. I hope that everyone's found it very useful, and we appreciate the questions that have been asked. If you do have any questions as a sort of follow-up that have not been touched upon in the Q&A, then please feel free to reach out to me. I can put my details back up in a moment, and we can circulate these along with the recordings if you want to share them with your sort of wider team. We also have a number of historic recordings which Tim has participated in, which touched on many other subjects which may be of interest, regulatory updates. So if you'd like to see those, they're in our resource section on the RSSL website. Keep an eye on our social media posts, and obviously feel free to follow us on LinkedIn. Just to mention that we also offer a range of technical training courses which are designed for professionals across the industry. We have several upcoming sessions over the coming months, including those detailed on the screen. I will put the slide back up in a moment, and they're scheduled throughout the year. So just as I say, check on the website for future events. And the part 2 of this event is going to be held in June. So just let me pop this slide back up. Ooh, and it's not moving. Ooh. Sorry, I don't know. I'm having a few technical difficulties. Sorry about that. I can't seem to move the screen, but yeah, I think anything else from anybody, or are we? Anything else, Tim? Are we all good?
No, we're all good. Hopefully, people got some pointers, and what we're doing the next webinar is kind of go from laboratory into the field. How do we really make sure these disinfectors work? How we're going to qualify them, and we'll kind of go through the whole validation exercise.
That sounds fascinating. That sounds really good. And we look forward to seeing you and hopefully some of the attendees next month. So thank you very much for your time today, Tim, and for all those that have joined the session this afternoon. Have a great rest of the day. Many thanks. Bye-bye. Bye.
Bye.
Bye.